So this assessment has a little bit of revision from our first half term, and then the other little bits about viruses, and I'll touch on membranes as well. So, multiple choice. With these, it's always good to start thinking about how to remove the distractors. Now with this, when you're thinking about distractors, you want to see if things are irrelevant or if they are factually incorrect. Now this question has 3 irrelevant comments. So increasing magnification would all be about changing the objective lenses. Removing excess steam, we use a little bit of filter paper and we can draw out. And killing a specimen is wholly irrelevant to the cover slip. Some specimens with a light microscope can be viewed alive as well. Next one down, we've got the knot. So again, thinking about your strategy, you can box the key operating part of that word. Now they've put it in bold. It's good practice to make sure that you are identifying these things. And then we've got Luca, so the last universal common ancestor from which all life present today is thought to have evolved from. Now, going through here, you've got one word, and it's this can. Now, this whole idea, can rather than have. And that's your kind of signposting in terms of your English language skills. But in terms of the science, you've got the biochemistry, the degenerate triplet, non-overlapping genetic code, and then these shared 20 amino acids. And then this last one is a bit more about the coevolution of viruses and thinking about the escape theory and things like that. Now, going into this one, not found in viruses. So viruses are not alive. They do not satisfy the kind of ringer or Mrs. Gren. So you think about reproduction, respiration, irritability or sensitivity, nutrition, growth, excretion, or movement. And then we also think about homeostasis as a key bit. And again, we're looking for that not a feature. So lots of enzymes such as reverse transcriptase found within our retroviruses. This is definitely true, and this is definitely true. Remember that capsules are bacteria. So you do need to focus in on those words. Here we've got a nice image that would have helped a few of you for the last question where you're asked to draw. So we've got our phospholipid bilayer with those phosphate heads pointing upwards. We've got glycoprotein acting maybe for cell adhesion or cell recognition. In here we've got a glycolipid, we've got a channel protein, we've got an extrinsic protein, and then we've got these bits which are like poorly drawn cholesterol molecules. So if you can identify cholesterol, there is only one correct one. If you're struggling and you don't quite know what this one is because it's a terrible drawing, look at what you can find and then you can start discounting things. So the glycolipid, we can go across and we can get rid of these two. Onto your protein, you've got number 3 being a channel protein, you've got a glycoprotein, where have you gone, like this one, and here, number 4, which kind of keeps us here. but we do have the glycoprotein. So we can discount this one. Number 3 is definitely not a glycoprotein. And then our last one, the phospholipid, hopefully you can all recognize that. So straight away we've used a process of elimination to work out what the correct response is. So phages are the ones which have the legs. This one is your typical virus structure, like an adenovirus. This is more like your viruses that infect plants, like a tobacco mosaic virus. And then this one, I don't have a clue what that's meant to look like. So, Miller array. And you've seen this question before, and hopefully much more of you are getting this correct, which is a nice thing. Now, we're looking for a limitation. So this is where we're looking for the negative. What was wrong with their experiment? Now, this is factually incorrect. This was none. They used that electric spark rather than ultraviolet radiation as the sole source of energy. They did run it for a number of weeks. They did have high levels of methane, and we believe that those are consistent with what they thought those early conditions were. Right. We do know that it should have been much higher in terms of the carbon dioxide concentration rather than just straightforward methane. So we have to think critically about these responses. Next one, we've got cholesterol, so we need to think about this regulation. So we've got a number of things here, and only one of them is relevant to cholesterol. So we've got hydrophilic channels, that's to do with proteins. Active transport, again protein, need to remember comparing that to enzymes. Cell adhesion, we think about glycoproteins. Then down in this one, fatty acids, making them likely candidates for those early ones. And this is where we need to think about that formation of the liposome, that sphere that was double membraned, double layered as a bilayer. Well, not double membrane. So, and this is your key word. Nice straightforward question about electron microscopes being compared with a light microscope. So we're comparing to say that the electron microscope has a lower magnification of a light microscope is wrong. Similarly, the resolution is higher. So every time we're seeing this lower, we can discount, and that process of elimination leads us to the correct response. Now, lytic pathways and lysogenic pathways, really important that you understand the two of them. And the key bit in here, with the lytic, we need to have the assembly then release. We need to have the replication of those virus units going through. Otherwise you end up with that lysogenic, the dormancy aspect. We've got a fluidity one, and this one is comparing, if you look here, and in here we've got the lower versus higher, and we remember that when we have a double bond, it kinks, and that will make it more fluid. So, correct answer here is A. The next one, the challenge of explaining. So we're thinking about like those limitations of the experiment and thinking about the evidence that we have. And the key bit of evidence is in this last statement of saying that they've not fossilised well, and we've got very little in the way of good quality, very old data. Like the strombolites from Australia, but even they're not at the beginning of life. Then into the next one, the escape theory. So this is all about the beginning, the origin of viruses, and the only thing that is relevant is here, this moving between those cells. The idea being that those units of genetic material start getting packaged up and being able to move between cells. A nice straightforward one. You're given a line, you're given a number, you've got the other number up here. All we need to do is convert this, if you want to go through the millimeters first and then into micrometers. There is no harm in just making sure you don't make a silly mistake. And then the last one, we've got a lysogenic. So this idea of this dormancy where it's not able to go through that lytic process. So the first one, which is wrong, is this because of the specificity, the idea of suppressing immune response, which is not correct. What is correct is this idea of the DNA not being transcribed, which then means it's not going to be translated, so it's just hanging about inside. It could be, the DNA could be copied and things like that, but it's not being transcribed, and then the following products are not being translated to make those capsids and the rest of the proteins needed for the viral structure. This question about the origin of eukaryotic is all linking back into how did membrane-bound organelles start, and this is where we need to look at chloroplasts and mitochondria. So the fact that they are membrane-bound, we pull this in. So that first marking point is for looking at this idea of endosymbiosis having a description, shall we say, of the evidence, and then pulling out what similarities they have. So it's a circular ring of DNA. They have their own ribosomes, so both double membraned or enveloped. Next one kind of pulls into that ribosomes about them being small. Most people got this one correct. So do use your images. So you've got your nuclear pore, you've got your nucleolus, you've got your free ribosomes, the cytoplasm. Use it to help and just go through. In here we've got, why is it not a plant cell? So what evidence do we have? So we don't have a cell wall, we don't have chloroplasts, we don't have a large permanent vacuole. It's trying to make sure that you're hitting all of those marks. Redundancy is a nice thing there, as long as you don't say something wrong, but you needed to state things that were present in that image. So you couldn't have had centrioles because they were not visible. Bit of endosymbiosis again, so we've got our ribosomes, we've got our genome, the genome not having introns, those non-coding regions within genes, and it is a naked ring of DNA. So we need two pieces of evidence. So we've got our ribosomes, and then we've explained about the DNA with detail. And then what do mitochondria do? Key bit here most people missed was aerobic respiration. A lot of people wrote down cellular respiration, which was not good enough. And then the last question, draw your fluid mosaic model. So you need 4 named membrane protein functions. So receptor, enzyme, channel, active transport. And then you're picking out the rest. So you need all of those things, and they must be labelled. Label lines need to be drawn with a ruler. You could have used the whole page, turning it around if you really wished.